Good morning, everyone. I'm Liz Fernandez, the training specialist with Professional People Training. And also with me today to assist is Kimberly Gish. She's a program specialist with Professional People Training. Just a few things before we begin. Please be sure to mute your phones. And also, we're going to hopefully keep everybody on mute throughout the presentation. And you can place questions in either the Q&A or the chat, and those questions will be answered at the end of the presentation. If you have any technical issues or any questions, you can also just send Kim or I a personal Teams message. So we have two speakers with us today. Dr. Catherine Duncan is an assistant professor in the veterinary pathobiology department at Oklahoma State University's College of Veterinary Medicine and co-director of the National Center for Veterinary Parasitology. She teaches parasitology in the veterinary curriculum and maintains a research lab that focuses on vector-borne diseases of domestic animals. She graduated from University of Tennessee's College of Veterinary Medicine, completed her residency in veterinary parasitology through NCVP, and obtained her PhD in biomedical sciences at Oklahoma State University. In 2022, Dr. Duncan became a diplomat of the American College of Veterinary Biologists, microbiologists, board certified in parasitology, and her most recent publications discuss ticks and tick-borne diseases of domestic animals. Also speaking today is Dr. Barrentos Moreo. Dr. Moreo is an IDEX resident in veterinary parasitology through the National Center for Veterinary Parasitology and is mentored by Dr. Catherine Duncan at Oklahoma State University College of Veterinary Medicine. At Oklahoma State University, he conducts research on vector-borne diseases and assists in teaching parasitology to the veterinary students. He earned his DVM in 2016 from the National University of Costa Rica and obtained his master's degree in microbiology in 2020 from the University of Costa Rica. Following this, he completed a residency in anatomic pathology at Auburn University and became a diplomat of the American College of Veterinary Pathologists in 2023. He completed his PhD at Auburn University in the spring of 2024, and his dissertation focused on serological and molecular diagnosis of heartworm in companion animals in the United States. His research interests are arthropod-borne diseases and infectious diseases in wild animals. And with that, I'm going to turn the webinar over to Dr. Duncan.
Thank you so much, Liz. And thank you all so much for tuning in. We're really excited to be here. Honestly, any chance to talk about ticks, we will take it. Hopefully you can feel that as we go through. And it is a big topic that we're going to talk about. And I realize an hour is not enough, but we're going to hit the basics so that we're all kind of having the same foundation of ticks. And then we're going to sprinkle in some more details and some recent literature and research. So hopefully, no matter what your background is with ticks, because I know some of you may work with ticks on a regular basis, I'm hoping that you can walk away and learn something today. So to get us started, to kind of convince you all that ticks really are important, I'm going to start with a case. And this was something that was referred to Oklahoma State when I was doing my residency here with Dr. Susan Little. So a dog came into a veterinary clinic and was covered in mats. And at first they just thought maybe it was a stray dog that had just been outside and developed these mats, and it likely could have been. But they started investigating all these mats of hair, and this is what they found, and they snatched a picture of this. So these are ticks at various stages of engorgement. When they went and looked at the dog's blanket that had been living in, you can also see all of these small, dark brown to black little structures, and those are all different life stages of ticks as well. So probably some immature stages that have fed, and then some of those bigger ticks are females that have fed. And so this dog ended up being infested with the brown dog tick, Rhipicephalus sanguineus. And it had so many of these ticks because it was living kind of in the environment where it was infested with these ticks, and all the different stages were feeding on the dog. The dog had actually become anemic because the ticks were taking so much of its blood, and they ended up having to do 2 different blood transfusions on this dog to get it back. And the veterinary technicians, they spent quite a bit of time, and they approximated about 10% of all the ticks that they saw on this dog. They cleaned them up and weighed them, and it was over 2 pounds of ticks. And so these are ticks full of that dog's blood. And so very quickly, an infestation, if it's not managed or identified, because in the mats, they couldn't the owners weren't able to see this, or the people that brought the dog in. And so you can see how quickly a tick infestation can really be a problem. And it's a reminder of, even though ticks have been around and we talk about them all the time, they're still a major issue for a lot of us. Another thing I want to draw everyone's attention to before we go into more details is that you may look at the slide and you may see just ticks. But what I see is I see all the differences. These are 4 pictures of 4 different species of ticks. And I look at this top left photo and I see the brown dog tick and I think loves dogs, love being indoors. So this is one that can have indoor infestation. I look at the picture on the top right, that's Ixodes scapularis. I'm thinking it likes the cooler months of the year. It transmits the Lyme disease agent. So I'm thinking of where it may have its greatest risk up in the Northeast or upper Midwest or along the Pacific coast. And then that bottom right photo, I see the Lone Star tick, excuse me, or Amblyom americanum. And that's a really popular tick where we are in Oklahoma, and it'll chase after you when you're out in the field collecting it. It's a very aggressive tick. And then in the bottom left tick, that's Dermacentor. I'm very fond of Dermacentor. It's what I did my PhD on. But I think of that tick as the summer-loving tick. And all of these transmit different disease agents. They have different host preferences. So it's hard for me to just see ticks when I look at this slide. I see so many differences between them. So hopefully by the time that we're done together in this hour, you'll be convinced that a tick is not just a tick. There's a lot of differences between them. And that's going to be important when you start going out looking for ticks or you're trying to prevent ticks. So today we're going to spend quite a bit of time on tick biology. I think there's a lot of interesting facts right there that we can share with you. And we can't go into all of the details, but at least some things. We're going to talk about some of the endemic and the exotic ticks that are in the United States. You know, we would need weeks at a time to go through all of them. So we've just selected a couple that have some interesting things we want to share. And then we're going to talk about how we go out and we survey for ticks and how we find ticks. The methods, we call it tick trapping, are unique to finding ticks. And so we'll go through those. So if you have any interest in that, hopefully then you're prepared to do that. And then we'll wrap up with just a couple points just to remember about tick prevention and control. So if you're having discussions with others about ticks, you'll be sure to be prepared to have that conversation. Now, like I said, I wanted to get us all on the same foundation because I'm not sure how much everyone knows about ticks or what the training is, the background is. So bear with me if you work with ticks on an everyday basis. I just want to get everybody on the same page. But taxonomically, ticks are within the arthropod group, and arthropod means jointed foot. So if you look at their legs, you do see those joints. And ticks are in the class with the arachnids, so they're not considered insects or true bugs. So if you want to be the most proper when you're talking about ticks, they are considered the arachnids. They're more closely related to mites than they are to lice or bugs or fleas. So just something to keep in mind when you're using common terms when you're talking about ticks. They're not insects. Now within ticks, we divide them into 2 main big groups. We have our hard ticks and our soft ticks. Our hard ticks are the ixodids. Those are the pictures on the top there. When you're looking down on a flat, unfed tick, which is what these are, you'll be able to see the mouth parts from that dorsal surface. Whereas the soft ticks or the argacid ticks, which are these bottom 2 photos, when you're looking down from the top, you will not see their mouth parts. They're more ventral on the underside. Another big differentiator, which is a little bit more obvious, is the presence or absence of a scutum. So those top 2 photos, I'm hoping you guys can see my mouse. You can see that these are 2 females, so they have partial scutums, or some people may call these shields. And so this is a hard chitinous structure that will not expand when they feed, versus with the females, that bottom portion will expand quite largely when they're feeding. Whereas the soft ticks, they do not have that scutum. Sorry, I heard a sound back. But they will not have that scutum. And so if you're trying to differentiate really quickly, you can look for that. But as ticks, even the hard ticks engorge, you know, they may start to feel really squishy and soft. But if you look closely, you'll still be able to see the scutum on those females. And that's how you're going to know that it's a hard tick. Now the tick life cycle, to go into that, we first need to talk about the tick stages. So there's 4 main stages of ticks. The egg's not pictured here, but we'll show it to you a little bit later on. But just think really small caviar is kind of what fish eggs, kind of what they look like, quite small, and they're usually in clusters. But the larva is going to hatch out of that egg. So that's the smaller structure there. That'll feed and molt and become a nymph. The nymph will also feed and molt and then will become up to an adult. And then it'll be either a female or a male. So they're not sexually differentiated between male and female until they become adults. Tick life cycles can become quite complicated. So I've hit the major ones that the ticks we're going to talk about. I'm going to go into those life cycles first. But for the hard ticks, there's 3 main cycles. On this slide, I'm showing you the 1 host and the 2 host. And the timing of the cycles can get a bit complicated. So depending on the environmental conditions, is it optimal for that tick survival? Do they have that host blood meal that they need to survive? That's all going to dictate how long these life cycles may take. If they have the perfect conditions and ample host, this can go rather quickly. If they don't have those things, they may take up to a few years. And there's a couple tick species that we know like to take a few years within a single life cycle. Now, the 1 host tick life cycle, I've always found this fascinating, but the larvae hatch out of the egg, they immediately find a host, and they stay buddies with that host until they're ready to fall back into the environment to lay the eggs, the females do. So the larva will attach, they'll feed, they'll molt to a nymph on the animal, they'll feed again, they will stop feeding, molt up to an adult, and then once the adults finish feeding and mate, then the females will drop off or the males will die, and they'll complete that life cycle. So on a single animal, you'll see all the different stages. Now, a 2 host life cycle is slightly different in that the larva, when they hatch out of the eggs, they will go and find a host, and on that host, they'll feed and molt to nymphs. So the larva in the nymph stage will feed on that same host, and then the nymph will drop off and molt into the environment and become an adult, and then that adult will find a host. So as the name implies, there's 2 main hosts involved in this life cycle. We in the US don't have ticks that do this 2 host tick life cycle, at least not the common ones. The common one that I pictured here in the bottom is hyaloma. And I wanted to put a picture on there because it's definitely a tick that we'd all should be aware of and on the lookout. It looks a lot like the amblyoma species, so longer mouth parts, brown body, but there's no ornate markings, so no white markings, and that'll be a big differentiator. There's also eyes present on either side of the scutum if you're looking at tick keys, and I can point those out when we start talking about tick morphology, but hyaloma is a perfect example of that 2 host tick life cycle. Now, the 3 host tick life cycle, it gets its own slide, mostly because most of the hard ticks that we have here in the US fall within this 3 host tick life cycle. So the larva hatches out as an egg, or the larva hatches out of the egg, it goes and finds a host, it'll feed to repletion, it'll fall to the environment, molt up to nymph. The nymph will go find a different host potentially, it'll feed. Once it's done, it'll go back to the environment, molt up to an adult, and then that adult will then go find another host. So within this cycle, there could be 3 different hosts. And so you can imagine the time in between these hosts, everything has to line up to have the perfect environmental conditions, has to find that host. And this is a life cycle that can really get slowed down. So some of the ticks that have this type of a life cycle could take a few years to complete. It also is a perfect example of how when the ticks is coming in contact with multiple different hosts, usually the ticks with 3 host tick life cycles are very great vectors of disease pathogens because they're coming in contact with a lot of different animals and they have more opportunities to potentially come in contact or pick up one of those tick-borne pathogens to then pass to another. So very common life cycle when we're thinking about ticks. The last cycle I want to talk about is a multi-host life cycle. This is the typical cycle for all of our soft ticks. So Atobius or Argus or Nithodorus are some examples. Now, the larvae are going to hatch out and they will go find a host. They'll feed, they'll drop off, they'll molt to nymphs. Now, a lot of the soft tick life cycles or the argacid tick life cycles, they have multiple nymph stages. So each time they're molting to a different nymph stage, they will find a different host and they'll fall from the environment to molt up to the next nymphal stage and find a new host. So you can see how this, again, life cycle could take a lot longer because they're looking for a lot of hosts through this nymph stage. And these soft ticks are unique in that they have multiple nymphal stages as well. But eventually, depending on the species, how many nymphal stages they have, eventually they'll become adults and that adult will go then find their final host, feed, and then mate and reproduce out in the environment. They always are mating out in the environment. And not all soft ticks actually are the adults even parasitic. Atobius is a perfect example. The adults have vestigial mouth parts, so they don't even need that blood meal to mate and then keep the life cycle going. So a lot of exceptions with all of these, but in general, those are the main life cycles. Before we can go and talk about the different ticks, and you can go out and look for ticks and identify ticks, you need to know a little bit about tick morphology. Now, this is the very basic of the tick morphology. If you go to tick keys like the one I have shown here, this is a USDA Ticks of Veterinary Importance from 1976. It's a great key to start with. It's one of my favorites. There's a lot of others out there, but this is always a good place to start. And they do a great job also defining all of the major features that you're going to be looking for on ticks, which is really great if you're first starting out and you're trying to learn tick morphology. But for the sake of this presentation and starting out with tick identification, there's a couple major areas on ticks. So ticks can be divided into their capitulum, so the more anterior region, and then their idiosoma, which is kind of their body. Now, on the capitulum, there's a certain couple areas that we'd want to be able to look at to help us identify. So that zoomed in picture in the top right corner is showing that capitulum, and we've got our mouth parts with the palps and the hypostome and the chelicerae. And then we also have our basis capitulum, and that shape of the basis capitulum can be very important, whether it's rectangular or triangular. That's going to help us with tick ID. And then also the mouth parts, the length of those mouth parts are going to be important. So if they're longer than the basis capitulum is wide, they're considered long mouth parts. And you can kind of group the ticks based on that. Now, if the mouth parts are not as long as the basis capitulum is wide, or just as long as it is wide, those are deemed short mouth parts. And again, you can kind of group the hard ticks into those two groups of long mouth parts and short mouth parts. Really good first area to look when you're doing tick ID. Kind of the second area that you're going to look on the idiosoma is the scutum. So the scutum may or may not have markings. They're typically white, but sometimes they can have an iridescent type look to them, especially if you're thinking about different tick species. And the presence of that and the markings themselves, the patterns, can help you make that ID. And this is a female, so she's going to have that partial scutum. So make sure mouth part length and then the ornamentation on the scutum and the basis capitulum are three just kind of basic things to always look at when we're looking at tick ID. And depending on the level of engorgement of a tick, you may need a magnifying glass or a dissecting microscope to look at this closer. Now, to differentiate the different life stages are also going to be important when you're thinking about tick ID and surveillance as well. You need to know what stages are present to be able to call it established versus reported. To differentiate between the immature stages, the larva and the nymph that we have pictured here, you can't always go on size. So the best thing is to look at the number of legs. So tick larvae, especially the hard ticks, they're going to have, well, all tick larvae, but here pictured as a hard tick, Amblyomma americanum, they're going to have 6 legs, whereas a nymph is going to have 8 legs. Now, adults have 8 legs as well. So to differentiate a nymph and adult, you need to look at the ventrum, and you need to look for the presence of a genital aperture or a genital pore is what a lot of people say. And it's usually positioned in between that last pair of legs on both males and females. On males, sometimes it's harder to see because it has a hood, but if you get rid of any debris and take a good look, it will definitely be there. It's not going to be nice and smooth. This picture is also a great example to point out where the anal pore is on ticks. So it's on the posterior region. And this is an Ixodes species. And so Ixodes is unique. It's a prostriate tick versus a lot of the others are the metastriates. But that arch is going to be above the anal pore. And that's how you're going to know that it's an Ixodes. A lot of times, unfortunately, sometimes it happens, depending on how the tick was removed, those mouth parts may have been damaged or have maybe stayed in the animal or the human. And so knowing for sure whether it's Ixodes, you can always flip it over, look at the ventrum, and see if this groove is above that anal pore. It's at least a good start, and then you may have to do some follow-up work as well. Now, to differentiate between males and females, we have a male on the left and a female on the right. It's all about the size of the scutum. So the males, the scutum goes almost all the way to the posterior edge. Most male ticks do need a blood meal, and so they only need a little bit of that space left to be able to engorge, whereas females have a partial scutum. It only goes about halfway down, and that's because she needs a lot of cuticle area to be able to expand while she's taking that blood meal. So that's how you differentiate the males and the females, and you can see how the ornamentation or the white markings on the scutum is going to be so much more obvious on a male tick. Going into really why we're all here is to talk about what ticks can do. And all ticks are parasites. They're actually obligatory parasites. The only food source they have is blood, all those that feed. And so they need to be able to get that food source. And now I'm going to hand it over to my colleague to take it away.
Hello, everybody. Dr. Barrantes Murillo over here. So I'm going to have a portion of this talk to talk about some generalities about the behavior during the feeding and the host questing by some ticks. Some concepts that are important to understand is that ticks are parasites, they need blood meals, and they need to attach to a host. So host recognition is very important process. They are able to identify several stimuli that can be chemical, like odors, which are the most important ones, and usually related to excreted substances from the potential host, including carbon dioxide, butyric and lactic acid, and also ammonia. Radiant heat from hosts also is another stimuli that can be detected by the ticks, and vibrations and colors. Colors, especially in shades, ticks are able to differentiate between the shade of a potential host versus the bright background from the sky. And in order to do that, they have several anatomical features that help them, but one is particularly relevant is the Haller's organ. The Haller's organ is, if you see the lower right image, is located in the tarsus of the first leg, and it usually has a pit and a posterior capsule. This organ is important because it's an olfactory organ, but also is able to detect radiating heat, like an infrared camera. The questing behavior is the definition in which the ticks expand or extend their forelegs anterolaterally toward a potential host. And they can remain in this position for a long time. And this questing usually influence the host specificities when they are located in different heights. If they are in tall grass, at the tip of the grass, like the picture in the upper right, probably they are looking for large hosts like cows. But otherwise, if they are questing on the ground, they are looking for small hosts like rodents. Moving forward to our next slide, when the tick finally finds a host, it will crawl over it and search for a potential sweet spot to bite, which it can take some time and attempts if the host does not reject the tick immediately or the tick doesn't drop off. They will look for the specific anatomical location. Different ticks have anatomical preference from the host, and they will cut the skin with the chelicerae and insert the hypostome. So actually, in the next slide, when we describe this tick attachment in more detail, if you see the left image, this is a TEM image of the capituli components of the tick. You can see how the palps are wide open, but the palps are not used for the feeding process. The chelicerae are a paired structures on the dorsal part that has terminal digits that are used for lacerate, and the hypostome is lying ventrally with a very narrow buccal canal. So we have this video to share to you to better make an example of these components and how they interact. So I want that you pay close attention to what is going to happen here. So we have the chelicerae that has these two terminal fingers or digits that will lacerate the epidermis. So the medial digits are larger and broad, and they can move laterally to make room to the hypostome. The hypostome has an harpoon shaped and has all these teeth that make it able to enter to the dermis until it finds the proper feeding pool for it. So moving on, describing these mouth parts are not the only sources that the ticks have to remain attached to the host. So within this first attachment, the ticks actually have an other strategies, and one important one is the secretion of the cementum. So the cementum is this proteinic substance that helps them to attach to the host. And if you see the lower left, that is a dermacenter tick, and all that material that is attached to the mouth parts, that is the cementum. So usually when you take out the tick, you think that you are grabbing the skin with it, but actually that is the cementum. So the cementum composition is of several amino acids, including glycine, serine, lysine, methionine, histidine, tyrosine. But the important thing here is that once this hypostome is embedded in the dermis, this secretion will start forming a cone. So in the right image, you can see how different species of ticks secretes different amounts of the cementum that are highlighted in the color orange. So just please notice how the cementum can form cones over the epidermis or surrounding the mouth parts, and also can extend laterally covering the epidermis, forming these lateral flanges. So these patterns are important, but the ultimate goal is the same, remain attached to the host. So if we move in to our next slide, I want to tell you a little bit about the process. Once that the tick is fixed, there's going to be a response from the host against the tick. So it will be anti-inflammatory, anticoagulant, an immunomodulatory response against a tick. So the tick has to have several mechanisms in which can elude this host response. They can either secrete through or regurgitate, will be the most properly term here, anti-inflammatory substances that they will be typically antagonists of histamine, serotonin, and bradykinin from the host, and also anticoagulant substances that usually inhibit the factor 10 and 5 that modulates the conversion from prothrombin to thrombin to maintain this feeding pool active. Ticks can markedly engorge. They can take up to 8.9 milliliters of blood, depending on the species. And usually the females have an increment at up to 120 times the original size, depending on the species. So if you see the image there, all this labyrinth structure that is highlighted with dark red is the midgut. And the arborizing white structures is the salivary glands. So the midgut is huge, is a huge component of the tick, and usually the blood pulls there. The hemoglobin has to be incorporated in the midgut cells through receptor-mediated endocytosis, and it will be metabolized inside phagolysosomes, liberating the heme group from the hemoglobin, which is toxic and has to be detoxified through hematine, and then storage in these hemodesmosomes, which is a special organelle for this. But all the amino acids and dipeptides that are the result after the metabolism of the hemoglobin, they will be transported into the hemolymph. However, despite this process, most of the proteins will remain on the midgut. So if we move on to our next slide, I will let Dr. Catherine explain this for you.
Thank you. I wanted to jump in and take the opportunity to talk about a study that was done here at Oklahoma State while we're talking about tick feeding. So this was done by Susan Little and Merck Animal Health, and they had designed the study to look at how an isoxazoline may affect the tick through maybe it's tick feeding before the tick actually dies, because that could be significant when we're thinking about passage of different pathogens. And Dr. Little designed it to compare between tick species. So she looked at Ixodes scapularis and Amblyomma americanum. I actually happened to be visiting as an extern at the time, so I got to help with this study, which is really great. And this is not peer reviewed, but it has been presented at a scientific conference, and we have permission to share this with you guys, but there's not a paper out yet. But what was really interesting about this, and what I wanted to share, is that through this study, we started to have some details about how we may have differences in feeding behaviors and phenologies between tick species that are both hard ticks. So when the study was designed, we infested dogs and then removed these live ticks at every 2-hour intervals. And we were assessing the ticks' motility and looking for early feeding evidence to see if it would change over time based if they had fed on a treated dog or an untreated dog. And so if we look at Ixodes scapularis first, when they were taken off those dogs very quickly, within a matter of a few hours, they actually were not able to right themselves. So we put them on their dorsum, and we're trying to a healthy tick very quickly, less than 30 seconds, will flip itself back over. Well, Ixodes scapularis, even though they weren't dead, they were still moving and were still reacting to our stimuli, our carbon dioxide. They were not able to right themselves. And so they weren't even able to exit this little tick arena that we created. Whereas Lone Star ticks, actually, over the same period of time that we gave them to feed, they were able to right themselves, and it was delayed. So those ticks that fed on treated dogs were having an effect, but it was nowhere near as severe as it was for Ixodes scapularis. Same with exiting that arena. It took them longer to exit if they had fed on a treated dog, but it wasn't as intensely of a response as Ixodes scapularis. And then when we were looking for maybe evidence in changes or disruption in the early feeding, in both tick species, the weight was measured, and we saw that if they were from treated dogs, they had a lower weight versus untreated dogs. But the engorgement index, so you can measure the scutal width and then compare the coxal, the fourth pair of coxal width, to try to determine how engorged a tick can be. We saw that Ixodes over time, if they had fed on a treated dog, then they weren't engorging as much. So some sort of disruption or whatnot is happening in that process well before the tick dies. Whereas in the Lone Star ticks, we did not see that disruption. So all in all, we thought this was a great opportunity to just be able to highlight how, even though, again, a tick is not a tick, even though we kind of think these are hard ticks and they feed in this way, there may be subtle differences that we can exploit and control when we're thinking about these ticks and how they feed. Thanks for letting me jump in.
So just wrapping up this part of the talk, a little bit over what is the reproduction and mating. So pigs usually have a large reproductive potential. They can produce up to 10,000 eggs, but there is important difference between the ticks types. For example, the prostriate Ixodes type ticks, they usually can mate on and off the host. But the metastriates, usually they mate only over the host. And once the male crawl into the host, the female will secrete sex attractants like dichlorophenol. So the male could start reaching her, and once the male has a close proximity to it, the female will secrete other substance like copulating attractants, like usually chloresteryl esters, in which the male will climb over it, allocate into the ventral part of the female, and then start probing the gonopore or the genital pore with the chelicerae. So once that it allocate that, the male will express the spermatophore containing the spermatozoa and place it inside the vulva of the female with the mouth parts. And this is a picture of two brown dog ticks copulating within the host. So moving forward to this process, which is closely related to the blood meal, it's important to understand that once that they molt from nymphs to adults, they need a blood meals, both males and females, to stimulate oogenesis and spermatogenesis. Then the females will not fully engorge if they are not inseminated. And after they inseminated, they will have a last big gulp. Um, these eggs of mass will be laid over several days or weeks. Usually, it depends on the secretion of a wax-like substance from the gene organs, and the females typically lose over 60% of the body weight, and they will die after ovipositing. So the number of eggs is proportional to the engorgement. Another important concepts here that we move in our next slide is how ticks are able to survive in extremely dry environments through a process in which they take atmospheric water from the hygroscopic salivary secretions. And ticks also can exhibit a behavior called diapause, in which allow them to overcome adverse environmental conditions. In this diapause, they stop feeding, they reduce the metabolic rates, and usually either the newly emerged larvae or the fresh molted ticks will wait until the optimal conditions to quest for a host and blood feed. And now I will pass over Dr. Duncan.
Thank you so much. So thinking about tick survival, the habitat that they live in is actually one of the most important things to allow them to survive. So when I think about the ideal tick environment, I picture this, and this is probably tick heaven for a lot of ticks, but there's actually certain ticks that survive in this or proliferate in this type of region or this type of landscape the best. And the Lone Star ticks, Amblyomma americanum or Ixodes scapularis, or Ixodes pacificus, those black-legged ticks, some people call them deer ticks, they love this more forest environment. So there's a lot of leaf litter. The sun, the tree canopy is kind of helping protect them from desiccation through the sun. There's good humidity. There's going to be a range of wildlife reservoir hosts coming through here, from small mammals to white-tailed deer. And this, if you're looking for these ticks in particular, this is the type of environment that you would want to target. This is also a picture of an ideal tick environment. We're thinking more of pastures and really tall grass. Now in this picture, it does a great job showing that forest edge. In particular for Dermacenter, that bottom tick picture, it really likes that forest edge because when it gets too warm, they can easily go over into the shade, hide under that leaf litter as well. But the Gulf Coast tick, Amblyomma maculatum, that middle tick picture, and then even Haemaphysalis longicornis, the longhorn tick, they really like these pastures and they can withstand a little bit higher temperatures and drier conditions than some of the others. Now, in the hottest part of the day, when they're wanting to hide, they will be at the base of the grass or crawl towards that forest edge or be on a host. So the timing of when you go out tick trapping or looking for ticks is also going to be really important. But this is another ideal tick environment when you're thinking about it. But what we often forget is that where there are tick reservoir hosts, such as white-tailed deer here or small mammals, it can become an ideal tick environment. This is someone's backyard. It's just a picture off the internet. And we always say wherever you're seeing white-tailed deer, there is the potential that you're going to see ticks. A colleague that's at Auburn always calls white-tailed deer seedy tick motels that move around. So it's the perfect chance a female feeds the repletion, falls off right in the backyard, and can start an infestation there, as long as the environment that's there and the temperatures that they have somewhere to hide out in the hottest part of the day or other hosts. So not just our ideal tick environments, don't forget about our backyards as well. But really why ticks, we're here to talk about ticks, is because what they do is parasites. Through that process of attachment and feeding, they can be very irritating to animals and cause quite a bit of pruritus and itching, and then secondary infections or alopecia because of that. When the ticks attach, they are creating a wound, and so sometimes we'll see secondary infections there. In really high infestations or severe infestations, particularly in those one host tick or ticks that can proliferate very quickly within an environment, kind of like the parthenogenetic longhorn tick, you can see anemia just because there's so many ticks feeding on a single animal. We see it often more on younger animals or those with comorbidities as well. Tick paralysis is another direct kind of condition that can come from ticks. So when they're feeding and they're regurgitating all those proteins, one may be a neurotoxin that will cause an ascending paralysis in animals that once that tick is removed, it will recede. But at first it can be quite scary. And certain ticks are more linked with causing tick paralysis, like Dermacentor species, but all of them could potentially do it. There's actually a case report not that long ago of a soft tick, a tobias, that is presumptively the cause of paralysis in a little mini horse. Production loss is also really important when we're thinking about ticks, especially for large animals, because they're so irritating. They can have cause a reduced weight gain on that animal because the animal is stressed or not eating as much. And especially if you're going to use that animal's hide for anything, if you have any of those ticks that are really aggressive feeders and create those big wounds. And then most infamously, when we think about ticks, we're thinking about how they transmit disease agents. And that's definitely something that we had to think about when we're thinking and talking about ticks. So this is not a complete or exhaustive list. It would take a lot of weeks to go through all of the different ticks that could potentially be on our domestic animals in the United States. But in large animals in general, I don't think they get as much attention sometimes with kind of the breadth and the diversity of ticks that they can find on them. So we decided to highlight a few of these. And I group them based on the life cycle that they have because it does help you know how to target them, how to go look for them, if that's your goal when it comes to tick research. So the one host ticks we have here, Dermacentor albopictus, I'm going to go into more details about that. But these one host ticks that we have listed, remember they come on as larvae and then they stay on that host through their life stage molts. And then as adults, once they're done feeding or done mating, then they'll go back to the environment or they'll die. And so it's really great when we're thinking about trying to control some of these ticks, especially if they have a high host preference, like some of these Rhipicephalus or Buophilus species. If they are very specific in who they want to feed on and they're one host ticks, it makes controlling them a little bit easier because we can target the whole population versus Dermacentor albopictus can be on a wide variety of wildlife hosts as well and a lot harder to kind of control. The multi-host ticks, those are our soft ticks. So Ornithodorus is one of potential concern on large animals, and I'll explain why. Atobius is the spinose ear tick. It does like to live in that ear canal, and so sometimes you'll see some different disease manifestations because of that, otitis externa or just head shaking. I've known horses that we've thought are colicking. They're throwing themselves down. They're so aggressive. They're so upset, and we look in their ears, and there's been some ticks. There was a case down in Auburn not that long ago, actually. And then three host ticks, there's a lot. Like I said, this is not the complete list, but just a couple highlights. Few different amblyomma species. Really in Oklahoma, probably the most common tick we find on animals that live outside is Amblyomma americanum, the Lone Star tick. Amblyomma maculatum and Pathogenesis, I'm going to go into more detail in a second. And then a couple Dermacentor species to be aware of. Dermacentor andersoni is the Rocky Mountain wood tick, so it's more often in those Rocky Mountain states. Dermacentor variabilis, through some recent research, there's always been a population in the eastern and the central US, but the research has shown that the population that's along that Pacific coast, it's been isolated for so long that it is now being considered a new species, a novel species, Dermacentor similis. So you'll probably hear that coming up a lot more. And Dermacentor variabilis is slowly moving its way that way. So as these two ticks start to interact more, it's going to be really interesting to understand, what type of hybrids could or could not be created. So stay tuned for more about how that's going to shift and change. Dermacentor nitens is the tropical horse tick, so it likes warmer temperatures, the tropical part, and it does prefer horses as well. It's one to kind of be aware of and know about because it is a potential vector of the agents of equine paraplasmosis. And so that's going to be important when you're tracking those foreign animal diseases. It is completely inornate, though. It has no markings. And when we get to Dermacentor albopictus, I'll kind of differentiate that and try to remember that Dermacentor nitens can look like that as well. And then Hemiphysalis longicornis is the newestly introduced tick to the US as far as we're currently aware, and we'll spend time talking about it. It's a very important tick to know about. There are some other Haemophilus that are in the US, and differentiating between them is going to be important. You sometimes can use host, but a lot of those tick keys, like the one I mentioned, may be able to help you differentiate between these. And then there's a tick that was introduced, or at least there's a case report from 2021, Haemophilus punctata, that can be on sheep that we also need to be able to differentiate from longicornis, and molecular methods are probably the best chance for that. They just transmit different disease agents. Ixodes, the two main species that we find on animals, there are a ton of Ixodes species out in North America, but they may or may not be found in high numbers on our domestic animals. So we'll talk about the importance of Ixodes as well. When I've selected a couple of these to talk about more, and how I decided to present these to you guys is as if the tick is introducing itself, let's say, as an icebreaker at a meeting. So what's the name, a fun fact about the tick, and then at least one point of significance. And if you're interested about any more details about these ticks or more details from that list, we can definitely talk about it at the end of the presentation, or I can help you give resources if you want more information. But a couple highlights. So this is Ornithodorus coriaceus, the pajuelo tick, and it's not talked about as much. It is a soft tick. It's got that miliated surface. It actually blends in with the environment that it loves the most, kind of arid, rocky regions. Its fun fact would be that it prefers to live at elevations of 600 to 8,000 feet, particularly in the western United States. I think ticks get so much attention in the Northeast and upper Midwest. And so I like highlighting any ticks that are out west as much as I can. But really important, significant point of this tick is it's been linked to as a possible vector of the Pajuelobacter abortivirus, a pathogen that may cause abortion in cattle. So the epizootic bovine abortion or foothill abortion. And so if a tick bites a 2 to 6 month old pregnant heifer, abortion may occur, or a weak calf at birth. And so this is just another important differential if you're in that region where this tick is present to have, and not a lot you can do ahead of time. So tick prevention and knowing where this tick is and targeting and looking for it is going to be the biggest thing when we're trying to control and give that education back to these producers. Now, Amblyomma cageninensis, or some of you may know this as Amblyomma mixtum. It's the cayenne tick. Its fun fact is that it's actually a complex or a group of at least 6 different species. So taxonomically, that's code for it's a bit of a mess, but we're figuring it out. And so this tick, I always like to show it too because it looks a lot like Amblyomma maculatum. So Amblyomma has these long mouth parts. They're longer than the basis capitulum is wide. They've got this really beautiful white ornate markings. And so if you want to differentiate between all of these, you've really got to look at the ventrum and kind of characterize and look at the different features on the different coxa. So the base of the leg where it attaches with this idiosoma. And there's, again, keys that go through this. But this is an important kind of complex or group of ticks to know about, particularly because back, I think it was about 2009, a horse down in Texas was diagnosed with Theileria equi, one of the agents of equine paraplasmosis. And they found a lot of these Amblyomma cageninensis ticks in the environment. And so they took some of these ticks that had fed on the positive animals, put them on naive, uninfected animals, and it was a successful transfer of the agent. And those horses, naive horses, did get sick once the agent was passed and they had antibodies. So it was determined that this tick is a competent vector for Theileria equi. And so one to be aware of and to know where it is and to track it for the purpose of tracking equine paraplasmosis. As far as I know, last I heard, it's really staying kind of in the southernmost tips of the United States. It will every once in a while be found in, let's say, Florida or the Gulf Coast states, but it really does stay down in Texas and lower. But if we're not looking at these ticks carefully and using really great tick keys or molecular methods, we may just be calling these Gulf Coast ticks and not taking a closer look. Now, this is our Gulf Coast tick, Amblyomma maculatum. Again, it has the long mouth parts and the ornate scutum. It is the cause, its fun fact would be that it causes gotch ear. Other ticks can cause gotch ear, but Gulf Coast tick really is the primary one. So this is an aggressive feeder. This is a tick that sometimes, even though it's secreting all different, regurgitating all of these different proteins to try to feed on us covertly, this is one that you can actually sometimes feel bite. It's a very aggressive biter, and because of that, it creates a lot of inflammation and potential edema and then long-term fibrosis. So you'll look out on a herd of animals and you can see the droopy ears because of the edema and the fibrosis. Of course, there's other reasons for that in cattle, but this tick is one of those. And then through experimental kind of infections and research, it is a potential vector for Ehrlichia ruminaceum, the agent of heartwater disease, which is an important one to keep track of, not endemic, widespread, very rarely found here in the US. But if it were here, or it was able to be spread outside of the areas where it's been diagnosed, knowing that Amblyomma maculatum, at least experimentally, can transmit it is an important thing when we're thinking about tracking this tick. You know, it is the Gulf Coast tick, but there are people up in Ohio that are studying this tick and it's fully established there. So it's moving around more than just at those Gulf Coast states. Now, Dermacentor albopictus is one of my favorites. It's the winter tick. So as the name applies, it likes those cooler temperatures. It's also a one-host tick. It's going to grab on as larva and stay on the whole life cycle. So you can imagine can get a pretty big infestation quickly. Its fun fact is that it comes in 2 different color variants. Now, if you go back to older tick keys, they will have these as 2 different names, but they did some interbreeding experiments and found that they can produce offspring. And so they've decided to clump them together, at least for now. So that top picture is what we call the brown or inornate variant. So there's going to be no white markings, whereas that bottom tick picture that's the ornate or kind of the white marking version of this tick. Now, Dermacentor nitens would look a lot like the tropical horse tick. That would look a lot like the inornate or brown variant of Dermacentor albopictus, but there are some pretty obvious features on the tick you can tell to differentiate them, as well as when they're out on host. So the tropical horse tick likes more the warmer time of the year, and then the winter tick is going to be out in the cooler time of the year. This tick is widespread across the US when you combine the 2 variants, and it's really important because it can come on in such major numbers and be so irritating. A lot of animals, particularly moose, they will rub a lot of their hair coat off because they're so irritated, and then it leads them to be very vulnerable to exposure, and they have died from secondary to infestations from this tick. Haemaphysalis longicornis, very, very important tick to know about. It's the newest introduced tick to the US. It's called the longhorn tick. It's completely brown in color. It's got short mouth parts, so shorter than the basis capitulum is wide. But the big differentiating factor for Haemaphysalis is that second palpal segment is flared. So that's what you're going to look for. Probably going to need at least a magnifying glass or zoom in with your cell phone when you're looking at this to see that flare. And then it can, in other countries, it can vector a variety of pathogens, but here in the US, at least for large animals, the biggest thing to know is it's a potential and competent vector for Theileria orientalis, in particular, kind of the newest emerging subtype Ikeda, at least in the US, that can be quite pathogenic, especially when it's newly introduced onto a farm. And so that's being tracked as the major vector, and we're seeing a lot more cases of that too. Where longhorn ticks are currently, this is the USDA situation report, the September report. It's now in 21 states plus DC. Just earlier this year, it was established in Oklahoma, so it's a hot topic around here. Everyone's talking about it. But they think it could have been introduced as early as 2010, maybe earliest. But really what started this biggest boom we're seeing right now is that it was 2017 at a sheep farm, a pretty bad infestation, and since then it is spreading. Now, this tick, as it was mentioned on the last slide, she can produce without a male, so the Pathogenesis, and that has allowed her to reproduce. And so we're seeing a lot of this tick in counties. They'll kind of spread out, and then the transport of animals, we're seeing why she's spreading so quickly is because she can reproduce so quickly. And they have very low host specificity. So they'll be found on a lot of different types of hosts, which makes her spread a lot quicker. And now that's starting to hit some of these states where we move animals around a lot, unfortunately, this tick will be pretty widespread across the US, and it's done it in a really incredible quick amount of time. Now, ixodes, we've always got to mention ixodes, mostly because of its public health concern. You know, large animals can be diagnosed with Borrelia burgdorferi, the causative agent of Lyme disease. But I always think about ixodes being really important, not just for the animals that we're taking care of, but the caretakers as well. There's a lot of disease agents, I'm not even going to the viral agents, that may or may not have an effect on the animals, but they could have an effect on the caretakers. And so keeping their health in mind can be really important. This tick's fun fact is that it does prefer cooler months of the year. So if you're looking for this tick or suspect this tick is around, you need to look and target those cooler months. Now, in the coldest parts of the year, they will go into diapause and hide under that leaf litter or the snow cover, but they'll be out at times you may not kind of expect. It's always a myth we're trying to bust. Ticks aren't just out in the summer. One thing I wanted to mention about ixodes, I get asked this a lot. You know, we have a lot of ixodes in the south. So on the right side of this map, the blue is where Ixodes scapularis is established, and the yellow is where Borrelia has been found. And you can see that this tick is slowly expanding across the different states. We do a lot of county surveillance in Oklahoma. There's probably, since this was published, probably more of those counties in Oklahoma should be blue. But you can see the big difference between the areas where ticks have had Borrelia identified and the states that haven't. And takeaway from this is that just because there's black-legged ticks present doesn't always mean Lyme disease is going to be there. And kind of a follow-up question that always comes after that is why? And we probably don't know the full why, but there's been a lot of research to look into this, and a couple hypotheses and explanations have come about. Now, within the species Ixodes scapularis, you can haplotype them and then put them into two different clades. So we have our American clade, which is mostly those ticks that are in the Northeast and Midwest, where we see a lot of Borrelia burgdorferi transmission. And then the Southern clade is mostly down in the Southeast, and that's where we're seeing kind of not as much Borrelia infected ticks. And when they started looking at these ticks, they noticed they have a lot of behavior differences, one of them being how they quest. So the American clade, they'll see on or above leaf litter. So maybe they're just coming in contact with animals that then we can identify, or we see disease because of the Borrelia. Whereas the Southern clade ticks may be below the leaf litter, and so they're just not coming in contact with the right host to either get infected or to pass that on to a larger mammal to then be able to identify that it's a disease agent. And then the biggest thing that I think a lot of people talk about is how the hosts that the Ixodes can pick is different in the Northeast and Midwest versus in the Southeast. So up where the American clade ticks in the Northeast and Midwest are, there's a much lower host diversity. So they don't have as many hosts to pick from. So they may be choosing a white-footed mouse more often, which is the reservoir for the Borrelia organisms. And so it's going to increase the chance of the tick to ingest that Borrelia and then pass it on to another host that may develop disease, versus in the Southeast where we have a lot of Southern clade Ixodes. There's a lot more host diversity in the ticks and the hosts that they could pick from, these ticks. And so think of it as a dilution factor. They've got a lot more to pick up. They're going to decrease their chance of coming in contact with any Borrelia reservoir hosts. We also have a lot more Ixodes that'll feed on lizards or other reptiles. And so those are not going to be our reservoirs for Borrelia. And so again, just dilutionally, the ticks are not going to be as likely, or they're decreasing that chance of having it. So may help explain why we see that difference in the North versus the South for Ixodes scapularis, but likely multifactorial of what's going on. Now we're going to finish on our tick on the Rhipicephalus buophilus. It's hard for me not to say buophilus. That was the original term, but Rhipicephalus is what it's currently considered. We've got two big species that we think about. Pictured here is Rhipicephalus microplus, because that male has that caudal protuberance. That's the southern cattle tick. And then the Rhipicephalus annulatus is the cattle tick, or the cattle fever tick. These are one host ticks. They grab on as larvae. They stay on those hosts. They do have a pretty strong host preference when they do get on that host. Its fun fact, and probably also its significant point, is that it's reportable, and there's a lot of effort, thanks to the USDA, for keeping this tick in control, under control. It's mostly considered eradicated from the US, except the most southern tip portions of Texas, and these maps may be a little bit outdated. But it's very significant and reportable because of what it can vector to cattle. So different Babesia species. If you look at the picture with the red blood cells, on the right, we have Babesia bigemina, or a large Babesia. And then on the left, the Babesia bovis, or the small Babesia. Both cause very significant disease in cattle, and is a main reason why we always want to know about this tick and be on the lookout for this tick. So that tick is a perfect example of why we do tick surveillance. And we do a lot of tick surveillance here in Oklahoma. There's a lot of different ways to do it. But if you kind of try to simplify it a little bit, you can break it into two types. You have passive, so you may have submission programs. Here we have a program called Show Us Your Ticks. It's ongoing and I can explain it if you're interested. But we're just asking for ticks from veterinary clinics, veterinarians, whoever gets onto our website to submit ticks. And there's a lot of these programs around. It's just great. We're pulling ticks off. Let's put them towards science. And then you can have active surveillance. So you're going out and actively looking for these ticks through dry ice traps, flagging, dragging, or even checking animals. And I'll describe some of these methods on the next slide. But when you're thinking about how you're going to actively go out, this guide by the CDC for environmental sampling, and they go into host sampling a little bit too, is such an incredible resource. If you have any interest into going out and looking for ticks, that is a great place to start. They go into a lot of detail, even how to create your flags and drags, the type of environmental conditions you need to think about when you're looking for ticks, and the different species of ticks. And it was just updated in 2020. So it does take into account Haemaphysalis longicornis, which is great. And then for host sampling, you can definitely look at domestic animals. I think it's always important to look for ticks on animals, but also looking at the wildlife reservoir hosts is a great way. Realize there's going to be some tick species on these reservoir hosts that may be specific to that wildlife host that we wouldn't see spill over to humans or domestic animals. So be aware of that, but there's still a really great place to be looking for ticks. And these are some of our methods. In that top left photo is a couple students who are out flagging. So they've got this piece of cloth that on a dowel rod, and this is really helpful in really brushy or woodsy areas. So you're just going to brush that in the vegetation, and any ticks that are out questing will grab onto that fabric, and then you can take those off with forceps and put into some ethanol. Of course, they've got their PPE on, which is great. And then drag, you would just take a larger piece of cloth with dowel rods on either end, and you would drag behind you, probably in more of a pasture type setting. Really hard to pull a drag in the woods. You can do it, but you're getting stuck a lot. So the flags are really handy. But the drags are great for picking up those species more on the pastures. And then the bottom right photo is showing a dry ice trap. So the dry ice is usually put on a center structure. This is how we do ours, at least. And it's emitting the carbon dioxide. The ticks are going to be attracted to that, crawl towards it, and that tape border actually has a sticky side up. And so then the ticks will stay stuck there, and you can go through after a couple hours and collect them. I learned recently that for longhorn tick, Haemaphysalis, they don't stay stuck on the tape very long through whatever mechanism they can get away. So if you're looking for Haemaphysalis longicornis, you need to check those traps more regularly to pull them off before they escape. Another thing I'm always looking for is making sure there's signs of maybe reservoir hosts, which some small rodent holes or homes, and then tracks of reservoir hosts is really great as well. So I'm going to finish on this slide just so that we can make sure we have a few questions if anybody's still hanging. But the drivers for tick populations, which is why one of the big reasons we're looking and surveying for ticks, is we want to know where they're going. And knowing where they're going, we need to know what is driving them to expand or just change where they are. And habitat's going to be a big part of that. What hosts do they have present? Where are those hosts moving? So what's causing those hosts to move? In the Western US, wildfires is a big cause of why these ticks may move around with their hosts. What's the lifestyle of the animals and their caretakers? We can certainly be carriers of ticks. And then climate is probably one of the most important things that's driving these tick populations. And when we're thinking ticks that are out in the environment, you know, they may be out at certain times of the year at the highest timing, and we call that peak activity or seasonality, and it's going to change depending on the tick species. But that's all determined by kind of the climate and what hosts are there. And when we think about seasonality, again, always trying to bust the myth that ticks are only out in the summer. This was a study done here in Oklahoma where they were looking every single week for ticks on horses, and you can see every month of the year they found ticks. Now in the summer months, they found, you know, the summer loving ticks, Emblem americanum, and in the cooler months of the year, they saw Dermacin or albopictus, the winter tick, and Ixodes. So we do find ticks every month of the year, even here in Oklahoma. So I think just for the sake of time, we will stop presenting here so we can take any questions. And I will go to both the chat and the Q&A. I'll start with the chat. Oh, sorry, there's quite a few here. Okay, Dr. Barrantes Murillo, do you want to, there's a question about cementum. Do you see that one?
Sure. So, to think about the cementum is to think about the application of it. It allows the tick to be attached to the host enough time. So the cementum production is driven by the feeding features of the arthropod that we are talking in this place. So it's important to say that you can find biological adhesives in several types of arthropods. The sandcastle worms will be an example of those. Not cementum properly, because cementum will be something specific from ticks. But also remember that not all the ticks produce cementum. Like some species of Ixodes, they don't produce cementum, and the argacid ticks or the Nutaliella, they do not produce cementum. So other arthropods can produce it, but the medically relevant ones, like ticks, lice or fleas or flies, it's unlikely that they produce it because the feeding patterns are very short, not long on the term. But sandcastle worms, they produce some kind of similar adhesive like the cementum.
Thank you. I think Alejandro has his hand up if you were hoping to come off mute. If not, that's okay. You can put it in the chat. There's another question asking about, are wild canids susceptible to Borrelia burgdorferi? And that's a great question. I haven't come across studies, characterizing the disease and describing that. But Borrelia burgdorferi has been found on a variety of different animals. Besides, it's very well characterized in domestic dogs and humans, but it's been found in cats and cattle and horses and etc. And so for it not to have some sort of infection model in wild canids I would be very shocked. Of course, wildlife are so incredible at adapting to disease and working through disease, especially parasitic diseases, way better than a lot of our domestic hosts. But it's a great question, and I can direct message you so that I can give you my email address and I can find some papers if there's anything out there, and there just might not be.
I believe that foxes has been affected by Borrelia. There are some surveys in North Carolina in foxes and wolves. They for sure found exposure, but I think foxes, they developed the infection.
Great, thank you. And then a question about how can ticks be stopped and killed? It's a great question. There's a lot of products on the market that can prevent ticks from even biting, so we'll call them repellents. The pyrethroids are a big group that's popular for that. We have a lot of other products that are more systemic, so they get into the bloodstream, and then when the ticks bite, then they'll ingest the compound and then they'll die. You also can do a few things environmentally to try to help. So on farms, there's patch burning to try to decrease the numbers of ticks. You can do what they call tick scaping, so that you may create a border of wood chips or gravel about 3 feet or more on the border around a barn or a kennel or a home per se. And it's quite a long distance for a tick to travel. But I will say that if they're on a wildlife reservoir host and there's no fence that's preventing that, then they can easily come back into an environment. And so tick control environmentally is actually very difficult and very frustrating. And so applying widespread acaricides is also very difficult. But I think that targeting the animal can be good. And then just looking for ticks is another thing that we can do. And then, yes, thank you, Liz, for putting our email addresses in there. If you guys have other questions, definitely Mary Kate, if you'll send me an email, that'd be great. And we can talk about that more if you want to get those papers about the fox, the wild canid infections. And then the last question, what should our VMOs be ready to share with producers about importance of tick control? Really great question. I think one of the biggest points is that there should be some tick control, or at least some tick surveillance or awareness is even the best thing. So if we're never looking for ticks, we can never know how to describe the risk. And so when we're thinking about it on farms, these pastured animals where there's constant reintroduction of ticks, especially for bringing in animals or if there's wildlife associated with that, you just have to know that you're going to find ticks and you can expect that there's a constant kind of source of ticks. So when they are working and handling the animals, doing those tick exams, knowing what tick species are present, so the tick identification will then help us build a list of what may be potential tick-borne disease risks could be there based on the species. Of course, there's a lot of great service. USDA APHIS offers the tick identification. So does a lot of veterinary diagnostic labs. We can always have more information. So getting them to help is really great. And then there are products that do work that they can apply to the animals. Like I said, the pyrethroids are a big group. Organophosphates, carbamates are also other groups. But what can be frustrating is that you have to reapply regularly, especially for ticks. And so if you know, again, what tick species you're dealing with, you can try to target it based on their peak activity. So when the risk is the greatest. So start with surveillance, look for the ticks. Once you know what ticks are there, then you can build really controlled plans around their peak activity and the lifestyle of those animals and what they're seeing. Question, is there additional research or interest in characterizing the geographic distribution of Ornithodoros species in the U.S. given concerns of African swine fever? Very great question. There's a researcher, Dr. Miriam Sala at Texas A&M, that has started working with Ornithodoros. As far as I knew, there was only 2 colonies, tick colonies, that are used experimentally in the US, and she just started the second one, at least at universities. And so she's looking a lot more into this because it is a very understudied area. And in particular, she was wanting to look for pathogens as well. So I will say there is interest, and stay tuned for any research that may be published. I think that may be the last question.
I don't see any in the Q&A.
Okay. Well, please reach out if you guys have anything. Really appreciate you hanging in. You know, we've been studying ticks for a few years, but we're always learning something else. So I really appreciate everybody putting in comments too, and happy to be here to talk ticks with you.
Thanks very much. Great presentation, great photos, learned a lot. So thank you again, and glad we finally got this off the plate and we got it done. So, all right, take care, everyone. Have a great afternoon.